Hello, everyone. My name is Simon, and I'm at Canada's Michael Smith Genome Sciences Centre. And thank you all for attending my talk. Today I'm going to be talking a little bit about some of the benchmarking work that we do here at the GSC. So let's get started. So first, just to ensure that we're all on the same page, I'm going to give a very, very high-level overview of the Illumina sequencing that we perform here at the GSC. A couple of months ago, my colleague Richard Corbett gave his Compute Canada talk, and I recommend looking at his presentation where he goes into much more detail. But Richard was very kind to let me repurpose his slide here for the purpose of my talk. So first off, we start off with the sample. Let's say we have a patient with colorectal cancer, and we have a colorectal cancer biopsy. And then from that biopsy, we can isolate the cells and then isolate the DNA. We shear the DNA and ligate sequencing adapters, and we put it through our sequencer. This is an Illumina machine. And the output from Illumina would be these short reads. Right now, we're at about 150 bases long. And we generate, for each run, generate on the order of billions of these short reads, about 150 long. Just to emphasize the point, in this example I just gave, we started off with a colorectal cancer sample, but it could just as well have been the matched normal. It could have been a biopsy from the adjacent normal healthy colon, and we would put it through the same process, and the reads that come would represent normal healthy colon. So billions and billions of these reads aren't actually very useful unless you align them to a reference genome. So here we have all our reads, and we align them to the reference, illustrated by this rectangle here. And you can see that these reads line up to specific portions of the reference, and that tells you where these reads belong. After the alignment, you end up with something called a binary alignment file, also known as a BAM file. This BAM file reports where the reference genome these reads align to. And again, from my previous example, if we started off with a cancer sample, then at the end of this pipeline, we'd have a BAM representing the cancer reads where they align. If we started off with a matched normal example, a sample of healthy normal colon, then we'd have a normal BAM. Here's an example of some real life alignments. This particular tool is called IGB and lets you visualize your alignments. So right now we're in a region on chromosome 15, and along the bottom here you see the different bases that make up the reference genome. And you can see that we're actually in an intronic region of this particular gene called OCA2. And in this part of the diagram, we have the reads that make up the BAM. And you see that with the exception of this part here, the reads actually agree, the bases of the reads agree with the reference genome, again, except for this part. So at this particular position, the reference genome contains an A. But as you can see here, all the reads at this position, instead of an A, have a C. And this single nucleotide variant, it's a single position, a single nucleotide variant, in this case has a phenotype in that the person that this particular sample came from likely has blue-green eyes instead of say, brown eyes. So this is an example, again, of single nucleotide variants. So what about somatic variants? What are somatic variants? So these are variations in DNA that occur after conception. So by variation, I don't necessarily, not just single nucleotide variants, but they could also be insertions and deletions, or indels for short. It could also be large-scale structural rearrangements in the genome. And these variations are not in germ cells, and thus are not passed on to future generations. So they're not in sperm or egg cells, but rather they're acquired, again, after conception. So why are somatic variants of interest? Well, they're interesting because some of them can actually act as cancer drivers. So here's an example of KRAS. There is a single nucleotide variant in KRAS that converts the amino acid G to D at the 12th amino acid position. And this is called a gain-of-function mutation because the resulting mutated KRAS is overactive and has been shown to drive and promote tumorigenesis in a variety of cancers, but specifically colorectal cancer. So somatic variants are variants that are in the tumor, but not the matched normal. So perhaps this person was a heavy smoker or had a very, very poor diet or some other environmental trait where he accumulated this particular mutation in life after conception. So what are the challenges of calling somatic variants? Well, there's the issue of sample purity. When a surgeon hands you a biopsy, that biopsy is unlikely to be 100% tumor. It's going to be a mix of some normal as well. And having that mixture of tumor and normal in the biopsy can potentially impact your the somatic variants that you get from your pipeline. Every technology has its own sequencing biases and errors, and of course there's alignment ambiguities. Your SNP prediction, your SNP prediction is only as good as your alignments, and of course there's also differences in the variant calling algorithms. So given all of these challenges of calling somatic variants, what are some ways that we can estimate the accuracy of a somatic variant calling pipeline? So one way to do this would be to construct a ground truth data set. So this ground truth data set would represent the variants you'd expect to see in your particular cancer sample. So if you're calling a variant that's not seen in your ground truth data set, then you likely have a false positive. That variant is likely not to be truly part of your cancer sample. Conversely, if you fail to call a variant that's in your ground truth set, and that probably means that your pipeline was unable to call a variant that actually exists, and that would be a false negative. And both false negatives and false positives impact the accuracy of calling somatic variants. So with this in mind, we had three goals for this particular project. For a particular cancer sample, we wanted to collect independent somatic variant data sets from different organizations. And from these independent somatic variant data sets, we curated a ground truth set of somatic SNVs and indels. Again, indels are abbreviation for insertions and deletions. And with this information, we can estimate the accuracy of the somatic variants that are called from a particular analysis pipeline. So the sample that we looked at is actually a melanoma cell line. It's called Colo 829. It's isolated from a 45-year-old male. And it's one of the few cell lines out there that actually has a matched normal. A lot of cell lines, you only have the cancer cell line and nothing else. But in the case of Colo 829, we do have a matched normal called Colo 829BL that was made from peripheral blood of the same patient. So here I'm showing 4 independent data sets of Colo 829. The first 3 will make up our ground truth data set. So the first data set comes from the Wellcome Trust Sanger Institute and was published by Pleasance and colleagues in 2010. You can tell from the publication date that this is from a while ago. So the reads back then were short, 75 base pair reads, and they sequenced the tumor normal to coverage about 40 to 32x. And one of the strengths of this particular study is not only did they provide a set of somatic variants, they also confirmed or validated a set of the somatic variants and somatic indels. So 497 somatic SNVs and 62 somatic indels by an independent method were confirmed to be indeed correct and true. The second data set comes from TGen, the Translational Genomics Research Institute. They're located in sunny Phoenix, Arizona. They sequenced with reads of 112 long and the tumor normal at coverages of 80x each. And the final set was from Complete Genomics, which a few years back was bought by the Beijing Genomics Institute as unpublished data, and they utilize a proprietary sequencing technology. So these 3 independent data sets that were done at different institutions, each with their own internal biases, with their different pipelines and different thresholds, constitute our ground truth, and we wanted to compare it to our own internal data set that we sequenced here at the Genome Sciences Centre, where we had 125 base pair reads, and the tumor normal were sequenced at a depth of about 100x each. So here we have a Venn diagram comparing the three data sets. Again, the study from the Sanger Institute, TGen, and Complete Genomics. First off, you'll notice that there is a large portion of somatic SNVs that were common to all three data sets. There's 26,713 at this particular intersection point. But you'll also notice that some of the other tools, specifically Complete Genomics, they'll call a wide variety of SNVs that are common only to that data set. And this isn't surprising because the Complete Genomics, again, they didn't use an Illumina platform and use their own proprietary sequencing method. So the numbers in the parentheses represent the independently validated SNVs that, again, came from the Pleasant study. So again, the 497 somatic SNVs and 62 somatic indels. And it's comforting to see that the majority, 433 of the 497 independently validated SNVs occurred in the intersection. And then we see 59 that are only seen in the Pleasant study and TGen result, and then 5 that are just unique to the Pleasant study. So to differentiate these independently verified somatic SNVs, we're going to call this intersection tentatively validated somatic SNVs. So called because they're not 100% confirmed to be true, but because they are all called by 3 different institutions using different technologies, they're very likely to be true. So they're not Sanger validated, they're tentatively validated. Here's a similar Venn diagram for the somatic indel data sets. Again, you'll notice that Complete calls a lot more complete, unique somatic indels. And you'll see that there's a lot fewer overlaps in somatic indels than there are in somatic SNVs. But again, you see that the majority of these independently validated somatic indels, thankfully, occur in the intersection of all 3 studies. So there's 53 that, of the 222 that were called, that were also independently validated. So again, this 222 set somatic indels that are called in all 3, we call them the tentatively validated somatic indels. So just as a quick review, we have a set of tentatively validated somatic SNVs and indels. These are the variants that should be called in our data set. Any variant that is not called would be a potential false negative. So if, for example, we fail to, in our own in-house data set, if we fail to call an SNV or an indel that is in this data set, that suggests that that's a false negative, that we missed that, we should have called it. Conversely, there's also the union set of somatic SNVs and indels, and these represent all possible variants that could be called. So going back to this Venn diagram, the union set would be all SNVs in all 3 of these independent sets. So those again, these represent possible indels that could be called. So any variant that we call that is not part of that data set, that's a potential false positive. So for example, if we call in our data set, if we call 10 SNVs that are not part of that union set, that likely means that the 10 that we have are potential false positives, and that would impact our specificity. So let's estimate our somatic SNV sensitivity. We ask the question, how many of the tentatively validated SNVs were called in our in-house somatic set? So you see here, in the blue we have our in-house data set, and in the red was the tentatively validated set. And all tentatively validated SNVs were called except for 51 of them. So these 51 were not found in our set. So that gives us a sensitivity estimate of over 99%. So we were able to call every single one of the 26,713 except for 51 of them. Somatic SNV specificity, we asked the question, well, how many of the in-house somatic SNVs were not seen in the union set of SNVs? So again, these are the ones that are likely false positives. So again, in the blue, we have our in-house data set. In the red, we have the union set. And you'll see that just under 3,800 of the SNVs that we identified from our pipeline were not seen in the union set. So these 3,791 represent likely false positives. So that gives us a false positive rate of about 9%, which means a specificity estimate of about 91%. With respect to indels, we're seeing, again, very similar trends. 31 of the tentatively validated somatic indels were not identified in our data set, and that gives us a sensitivity estimate of about 86%. And there were 160 somatic indels in our data set that were not found in the union set of indels, which again are likely false positives. So that gives us a false positive rate of about 23%, or a specificity rate of just under 77%. So with this constructed ground truth for Colo 829, we were able to estimate the sensitivity and the specificity of our somatic predictions. But that doesn't end there. This particular ground truth set can also be used for other things. For example, to benchmark publicly available somatic SNV and indel prediction programs, which I'll go over in the next couple of slides. So in our pipeline, we have a variety of different tools that are in the literature, published and publicly available. And before choosing a particular tool to use in the pipeline, we have to do a validation or a benchmarking process to confirm that this particular tool is indeed the best for our needs. So the first step is to do a literature search. So we queried BioStars, PubMed, or even plain old Google for somatic callers, and we identified 28 such tools. And then we have to list some requirements, things that we need for the tools to be able to do. So the first step is just it has to be published in a peer-reviewed journal. The tool should be maintained, so someone should be providing bug fixes and updates, and the tool should be supported. So, for example, the authors respond to questions on BioStars. And the tool should output SNVs and indels in variant call format, or VCF. This VCF format is kind of the standard in the genomics and bioinformatics community, a standard on how to share variant data. So if a tool does not output in VCF, then that's an extra step that we have to do in converting them to VCF format. So that was a requirement for us. And of the original 28 tools, only 11 met these 4 requirements. So we continued on with these 11 tools. And just to emphasize, this is the strategy I just described is a common strategy for benchmarking any tool, not just SNV callers. So we perform a literature search. We do a local installation of the appropriate tools. We construct a ground truth set. So we're trying to answer the age-old question as to what is truth. And we take this ground truth set and we run various tools on that ground truth set and compare results. Compare results with respect to the criteria that we set up. And eventually we choose the tool, or tools, plural, to use. And once every few years, as new tools become available, new versions get released, we reevaluate and, if necessary, update tools or specific version. So I'm going to just quickly go over some of the SNV callers that we identified. So there's a tool called LowFreq from the Genome Institute of Singapore, Mutech from Broad, Shimmer from the NIH, Freebase from Boston College, Platypus from the Wellcome Trust Center for Human Genetics, good old SAM tools from the Sanger Institute and Broad. Somatic Sniper and VARScan 2 are both from Washington University in St. Louis. RTG Somatic actually comes from a private company, Real Time Genomics. Strelka comes from Illumina, and also a local tool that we developed at the BC Cancer Research Center called Mutations Seek. So for each of the somatic callers, we want to run it on our in-house tumor normal Colo 829 sample. And for each of the somatic callers, we wanted to report the wall clock run times, just how long did it take to finish, running in the same environment, number of somatic SNVs that were identified, and estimate the sensitivity and specificity. Again, all of this is possible because we've curated a ground truth data set. So here I'm showing the runtimes for the 11 tools. Along the x-axis I have, again, the 11 tools, and then on the y-axis is the number of hours. You'll notice that there are some blue bars. The blue bars represent tools that let you split your variant calling into different jobs based off of chromosomes. and the tools that are listed as red did not allow that. You could only run this as a single job. So this is just more for parallelization. Theoretically, the SNVs that are on chromosome 2 should be completely independent of the SNVs on chromosome 1. Thus, you can split each of the jobs based on chromosome and then run them separately, as opposed to just run as one batch, which unfortunately these tools in red don't allow. You'll see that Strelka is purple. It's purple because it's neither blue nor red. It's a bit of both. Strelka is nice in that it performs the parallelization for you. You just submit the one job, and it will take care of the background computation and split as necessary. The main thing I just wanted to highlight from this particular plot, you can see that for the most part, the tools run just under a day or so, with the obvious exception of VARScan 2, which actually took over 80 hours to run. In this plot, I'm showing the number of somatic SNVs that were identified. Along the x-axis, I have again the 11 tools, and along the y-axis, I have the counts. And in this particular plot, I want to emphasize that this particular tool from NIH called Tumor actually identified 160,000 SNVs, whereas a few other tools identified just under 40,000 or so. Low-Freq, Mutech, Mutation-Seq just a bit more than that, and Strelka. There was a recent study from the Broad Institute that wanted to estimate the variability in the number of somatic SNVs that you'd expect from a melanoma sample. And on the low end, that was you'd expect about 3,000 somatic SNVs. On the upper end, it would be just over 100,000 somatic SNVs. So right away, that suggests there's quite a few false positives in this shimmer run, because we're already well over 100,000 and in fact, over 160,000. So here we want to compare the sensitivity versus the specificity of all the somatic SNV callers along the x-axis. We have the specificity estimate from 0 all the way up to 100. And on the y-axis, we have the sensitivity estimate. You'll notice that the sensitivity estimate is much narrower, just from 97 up to 100. Whereas, again, it's much wider. You see a much wider range of specificities. And you can see Shimmer right here. Again, it had really good sensitivity, well over 99.5%, but its specificity left something to be desired. It was actually quite low, about 25%. In this particular plot, the closer you are to the upper right-hand corner, the better you are. And in that case, we see that Strelka and Mutation-Seq, which actually had the greatest sensitivity, performed quite well compared to the cluster of tools here and a smaller cluster over here. So based on these results and some other ones which I haven't described, we decided for our pipeline to use Mutation-Seq and Strelka. I touched on this briefly earlier, but again, the biopsy that you receive from a surgeon isn't going to be 100% tumor. In fact, it'll probably be more like 60% tumor or maybe 50% tumor. So we wanted to ask the question, how does the tumor content of a biopsy impact the sensitivity and specificity of predicting somatic SNVs and indels? So the lower your tumor content, the more likely you're going to miss somatic SNVs and indels. So we want to know, how low can you go in your tumor content before you start missing a significant number of somatic SNVs and indels? Like if a surgeon provided you a biopsy that was only 10% tumor content, is it still usable? Or do we go back to the surgeon and say, no, we can't get any useful data from this particular sample? So once again, using the ground truth COL829 data set that we curated, we performed a bioinformatics titration of our in-house COL829 tumor normal sample of both the COL829 BAM as well as the matched normal COL829 BL BAM. So this slide is a little bit busy, so let me walk you through it carefully and slowly. Here we have our original tumor BAM, and we're going to call that 100% tumor content. This is the full BAM that we got from the cancer sample. And then we downsample it to 90%. So we randomly choose 90% of the reads that were in the original BAM file, and we call that a 90% tumor BAM. We take that resulting 90% BAM and we downsample it some more until it's 80% of the reads. And then we take that 80% BAM and downsample it again down to 70%. And we go on and on and on all the way until you have a BAM that's only 10% of the original tumor content. So we go from 100%, which is again the full untouched BAM, all the way down to 10%. And if you do the same for the normal BAM, the Colo 829BL BAM, so we have 100% that's fully normal. We downsample such that we only have 90% of the original reads, and we downsample some more all the way until we get to the 10% BAM that only has 10% of the normal reads. And then you can see the little addition signs here. We add them together. So you take the 90% tumor content BAM and add it to the, you merge it with the 10% normal BAM. So you can see that in this resulting BAM, you'll have 90% of the reads that come from the tumor sample, but 10% of the reads come from the normal. And then you do the same for the other BAMs, 80 and 20, 70 and 30, 60 and 40, all the way down to 10% and 90%. So in this particular BAM, we only have 10% tumor reads, and the rest of the 90% represent reads from the normal sample. And you take all of these resulting BAMs and you feed them into the somatic variant, our somatic variant pipeline, to get an estimate as to how a low tumor content can impact somatic SNV and indel prediction. You'd probably have the most somatic SNVs and indels predicted at the full tumor content BAM, but as you go down, you'd probably see fewer and fewer. So here's a plot showing on the x-axis I have the tumor content from 10% up to 100%. On the y-axis, it's just the sensitivity from 0 all the way up to 100. All the blue lines represent the Sanger-validated SNVs and indels. Again, these were the ones that were independently confirmed by the study by Pleasance et al. That, again, is represented by the blue lines. All circles represent SNVs. All triangles represent indels. So you see that once you have a plot like this one here, you can start answering some pretty interesting questions. So one question you might ask is, well, what's the minimum tumor content in which I can expect to get about 95% of the SNVs I'd expect to see that should be there? So you look at your plot. This is again the SNV line, and at 95% sensitivity, you can see that we can get it at a 25% tumor content. So even at a tumor content as low as 25%, you'd expect us to get about 95% of the expected somatic SNVs that you should be getting. You can ask that same question in a slightly different way, the opposite way. So, for example, if a surgeon provides you with a sample with a tumor content of 40%, well, what percentage of the SNVs do you expect for you to be able to call with a tumor content that low? Well, actually, you actually call it quite a bit. It's actually greater than 99%. At a tumor content of 40%, you're actually calling well over 90% of the expected SNVs. Indels, not so much. This probably highlights how difficult it is to accurately call somatic indels. Even at a tumor content of 100%, we're only able to call about 86% of the somatic indels we'd expect to see. We did some similar work. I won't go into detail of this one, but this is very similar to the previous slide, except instead of small mutations, instead of SNVs or indels, these are more large-scale structural variants, which would include translocations, large deletions, duplications, and inversions. You can see that our particular in-house tool does somewhat poorly at lower tumor content, but gradually increases in percent identified as your tumor content increases. Whereas this external tool, DELI, actually performs kind of very similarly at tumor content 30% up to about 80%, but then increases a bit with increased tumor content. This isn't to say that, you know, in our pipelines we're missing some 40% of expected structural variants. We have various other pipelines can help recover most of those variants that are missed by these two particular tools. I just wanted to mention that a portion of this work was published last year in scientific reports with our colleagues at Illumina and at TGen, and this is the particular reference here. And for the community, we've provided a reference standard for cancer genome sequencing, specifically COL0829. You might know of the Genome in a Bottle Consortium, which provided high confidence ground truth set for an individual in A128A in the 1000 Genome Project, but that isn't a cancer sample. We provided a somatic reference standard for cancer sequencing. And just to emphasize that when it comes to evaluations, this is the standard protocol that we follow. We perform a literature search, local installation of the tools, you construct a ground truth data set, and you define what is truth. You define what results you'd expect to see based on evaluation criteria, and from that you choose the tool that you use. So these are following that previous protocol. These are some of the key tools that we use in our production pipeline. So for alignment, DNA, BWMM, RNA, we're using an in-house tool called Jaguar, eventually switching to STAR from Cold Spring Harbor. Bisulfite sequencing, Novoline, SAMTools, MPileup for single sample SNV and indel calling, CNV, a particular tool called CNA-seq, ApolloH for LOH prediction, structural variants for genomes, as shown in the previous slide, Abyss, in-house tool, Delhi, Manta. For transcriptomes, predicting structural variants for transcriptomes, Transabyss, Diffuse. Also currently evaluating a tool called Chimera Scan. For ChIP-seq, we're using an in-house tool called FindPeaks, but we're likely switching that out to the community standard, this tool called Max2, but we're just doing some further evaluations on this particular tool. So in conclusion, some of the key takeaway points I'd like to leave you with is that we were able to construct a ground truth data set of somatic SNVs and indels for this melanoma cell line, COL0829. And with that ground truth data set, you can do some pretty interesting things, such as estimate the sensitivity and specificity of your own pipeline. You can use that ground truth set to benchmark various tools. In our case, we benchmark some somatic SNV callers and compare their runtimes, number of somatic SNVs called, as well as their accuracies. And we even used the ground truth set to say something about low tumor content samples. And that even at, say, low tumor contents, tumor contents as low as about 20%, we're still able to extract out a significant number of somatic SNVs and indels. Lots of people contributed in many ways to this work. I'm not going to name everybody, but this work was basically done with 3 institutions. Again, Canada's Michael Smith Genome Sciences Centre, TGen, and Illumina. Okay, thank you very much.